Welcome to FinStex from Laura and Anthony.
Hello, everyone.
Get ready for an absolutely mind-blowing story we're unpacking today. It involves one single individual, a whole lot of venomous snakes,
Hmm.
and a truly revolutionary possibility, really, in the world of anti-venom.
Indeed, this is a narrative that really grabbed us from the start. It's quite an extraordinary example of how one person's very, let's say, unorthodox journey
You could say that.
has bumped right into the cutting edge of scientific discovery, leading to something potentially life-changing.
Okay, let's get into it then. The central figure here is Tim Fried, a snake enthusiast based in Wisconsin.
Right.
For a staggering 18 years, he undertook what you can only describe as an extreme personal experiment, injecting himself with snake venom.
And not just injecting.
No, and incredibly, allowing himself to be bitten. Can you even imagine?
It's hard to wrap your head around.
We're talking about over 200 venomous snake bites and more than 600 venom
It's
injections.
just staggering numbers.
And not just, mildly venomous snakes. We're talking cobras, mambas, taipans, the really dangerous ones.
Yeah.
The sheer audacity, the scale of this is, well, it's hard to fathom.
Yeah, and what immediately sparks curiosity is the why, isn't it? Why undertake something so perilous?
Exactly.
From what we've gathered, it seems to have sprung from a deep personal fascination with these creatures,
Okay.
coupled with a very real desire to make a difference for snakebite victims worldwide.
So kind of a mix.
It's a really unique combination, personal interest taken to an absolute extreme, but also a strong sense of altruism driving it.
And the results, the results of this nearly 2-decade-long self-experiment are just phenomenal.
Mm.
Fridley's body essentially turned into a kind of super producer of antibodies. It developed an incredibly broad and potent defense against a huge range of venoms.
That's the key part.
Yeah. Here's where it gets truly fascinating. His system achieved a level of, broad spectrum of immunity that current antivenoms, the ones made in animals,
Right, the traditional way.
they haven't been able to match that.
That's a key point to underscore, definitely. Think of antibodies like specialized keys designed to fit specific locks, the locks being components in the snake venom.
Okay, good analogy.
What made Fried's situation so remarkable, apparently, is that his body started generating some of these master key antibodies, keys capable of neutralizing venoms from a whole wide variety of snake species.
Wow.
So, recognizing the profound implications of this, immunologist Jacob Glanville and Professor Peter Kwong, they led the research to investigate this unique resource.
Right, digging into his blood samples.
Exactly. Their work focused on identifying and then isolating two particularly effective, broadly neutralizing antibodies from Fried's blood.
And these weren't just, identified and put in a database somewhere, right? The researchers went a step further.
They did. They combined these powerful antibodies with an enzyme inhibitor called verisplatin.
Verisplatin.
Okay. Yeah, to create what looks like a significantly more effective antivenom. Now, when we say enzyme inhibitor, you can think of it as something that steps in to block other harmful actions of the venom.
So, another layer of defense.
Precisely. Blocking different pathways the venom uses to cause damage.
Got it.
And the early findings from laboratory tests are genuinely exciting. Really promising stuff.
So what did they find?
Well, this novel antivenom demonstrated the ability to shield mice from lethal doses of venom from an impressive 19 different snake species.
19. That's huge.
It is. And this included some of the most dangerous snakes out there. King cobras, mambas, taipans.
The big ones.
Yeah. To really appreciate this, you have to remember that existing antivenoms typically offer protection against a much narrower spectrum, often just specific to certain regions or snake families.
So this is potentially a massive leap forward.
It really could be.
Okay, so this is incredibly promising, obviously. But looking ahead, what are the crucial next steps? And what limitations do we still need to keep in mind here?
Right. Well, if you connect this to the typical journey of, say, drug development,
Mm-hmm.
these positive results in mice are a significant first hurdle cleared, a great sign.
Mm-hmm.
However, the next critical phase involves human trials. You absolutely need those to confirm safety and efficacy in people. That's non-negotiable.
Of course.
Furthermore, the current evidence suggests that this new antivenom shows the most robust activity against the venoms of elapid snakes.
Elapids. So that's the cobras, mambas, kraits.
Exactly that group. Viper bites, which unfortunately account for a large proportion of snakebite deaths globally.
Yeah, a huge problem.
They're currently less effectively addressed by this specific formulation. So it's not quite a one-size-fits-all solution just yet.
Okay, that's an important caveat, but the scientific work is continuing, isn't it? I mean, what are the ongoing efforts to broaden the effectiveness and maybe move closer to a truly universal treatment?
Absolutely, the work doesn't stop here. Researchers are actively working on strategies to expand the range of venoms that this antivenom can neutralize.
Focusing on those vipers, presumably?
It's exactly, with a particular focus on including vipers more effectively. Optimizing the dosage and how the treatment is administered are also vital areas of research that are underway right now.
Makes sense.
The crucial advancement here, though, is that having identified these master key antibodies from Freed, future progress can now be built upon through rigorous scientific investigation, in controlled laboratory settings, safely.
Right, using the information gained, but without needing anyone else to take those kinds of risks.
Precisely. The foundation is there now for standard scientific development.
Ultimately, then, despite its very, very unusual beginnings, the potential impact of this research is just immense, isn't it?
It really is. A truly universal snakebite treatment could prevent tens of thousands of deaths and disabilities worldwide each year. It's a staggering number.
It's hard to even comprehend that scale.
Exactly. The identification of these broadly neutralizing antibodies, coming from someone who undertook such an extreme personal risk, it represents a remarkable, if unconventional, contribution.
Yeah.
It provides a tangible pathway toward a future where a snakebite perhaps no longer carries the devastating threat it currently does for so many communities globally.
So just to kind of wrap it up, we've looked into this extraordinary story of Tim Fried, whose 18 years of self-immunization against snake venom, a huge personal risk led to the discovery of these unique antibodies.
Mm.
These antibodies have, in turn, enabled scientists to develop a highly promising new anti-venom, one that has the potential to be significantly more effective and cover a broader range of snake venoms than our current treatments.
It's a truly compelling intersection of, well, extreme individual initiative and cutting-edge scientific breakthrough.
Yeah. What's particularly thought-provoking here, I think, is the idea that such a seemingly unconventional and, let's face it, incredibly high-risk path
Definitely high risk.
could yield such a significant scientific advancement. It really makes you consider where unexpected breakthroughs can sometimes originate, doesn't it?
Nice. It certainly does. It leaves you pondering that relationship,
Yeah.
between individual drive, maybe even obsession, and the rigorous, methodical processes of scientific research. It's a fascinating tension.
It really is, and definitely food for thought about the balance between individual initiative and scientific rigor, especially when you're dealing with such extreme risks.
Indeed. And for anyone listening who's interested, there are numerous avenues to explore further, learning more about antibody research itself or the ongoing efforts in anti-venom development globally. It's a really active field.
Well, that's our time for today. Don't forget to like, share, and subscribe.